Yes, sir. We are live now, sir. Good morning, everyone. Myself, Dr. Sarfaraz Kamboli. I would like to take this opportunity to host this event and welcome you all the participants in one-day seminar on QBD in pre-formulation study. Dear friends, goal of QBD is to achieve meaningful product quality specifications that are based on clinical performance, also to increase process capability and reduce product variability. So for this, we have a very eminent personality with all of us. I would like to give warmest welcome to Dr. Mahapare sir, Associate Professor, Government College of Pharmacy, Chhatrapati Sambhajinagar. Welcome, sir.
Yes, sir. Thank you, sir.
Completed his graduation from Government College of Pharmacy, Karad, and post-graduation in pharmaceutics from NDMVP Samaj College of Pharmacy, Nashik, and had been awarded doctoral degree from Shivaji University, Kolhapur. He has more than 22 years of teaching and 14 years of research experience. He is a recognized PG teacher and PhD guide. He is approved examiners of various universities. He is actively involved in the field of research. He has received research grant of rupees 4.20 lakhs. He has received Best Research Paper Award from Indian Drug Manufacturers Association for his research paper in Indian drugs. He has published more than 45 research papers in national or international journals and authored 3 books and one book chapter. He has guided more than 40 postgraduate students. He has delivered talk as a resource person in various conferences. He is a life member of Association of Pharmaceutical Teachers of India. So today we have such an eminent personality. Now I request Dr. Amer Kaji sir, Program Coordinator for this today's seminar, to give a brief agenda of the seminar. I think sir has some technical issues. So before starting session, I request all the participants, if you have any questions, please type them into your chat box in your control panel. Now I request Dr. Mahapare sir to start the session. Please sir, continue.
Okay. Thank you, Dr. Sarfaraj, for giving my introduction. And thanks to KT Patil College of Pharmacy for inviting me for delivering the session on quality by design in pharmaceuticals. Yes, please confirm whether it is visible or not.
Yes sir, it is confirmed sir. It is visible
Okay.
sir. Yes.
So please make a panel alignment. Okay, so thank you.
Okay sir.
So, dear participants, I would like to welcome all of you for the session on quality by design. In short, it is QBD in pharmaceuticals. So, quality by design, this concept is everywhere in all the sectors, we can say. So, here particularly, we are going to talk on quality by design in pharmaceuticals. So, the points we are going to see the introduction. What is the current approach? Quality by testing approach. What are the advantages of QBD? Current versus QBD approach to pharmaceutical development. What is the benefit to FDA and industry of employing this QBD? Overview of QBD. Elements of QBD. Design space, concept of design space, tools of QBD. DOE. And if the time permits, we'll go for one case study. Now, what is quality by design? So, quality by design means designing and developing a product and its associated manufacturing process, and that will be used during product development, and it should ensure you that product consistently attain its predefined quality at the end of the manufacturing process. So designing and developing a product and its associated manufacturing process. So the product which will make, so some predefined objectives are there in the mind, so that the disintegration time of the tablet, hardness of the tablet, dissolution of the tablet, so the limit which you have set, and how we can attain that quality at the end of the manufacturing process. So the designing for that, it is nothing but a quality by design. So in short, what is quality? So quality in short, in layman language also you can say, this means which gives satisfaction to consumer or customer. So if you'll go and will purchase, mm, you will go for purchasing of the television. So in market, number of brands are there. So we used to prefer Sony, LG, because the quality. So now that is, it gives satisfaction. Another thing is that, what do you mean by quality? So now that is, it should be affordable to you. Say for example, you'll make any tablet. So in the market, tablet is available per strip 20 rupees, 100 rupees, and 500 rupees. So if you cannot afford 500 rupees tablet and the same quality which you, if you can get with rupees 100, so the limit which you have set, and if you're getting the quality above that, so now that is definitely you can go for the purchasing of rupees 100. There is no need to go for rupees 500 per strip. So quality should be affordable. It should give satisfaction to you. So this approach QBD, it is a systematic, holistic, and proactive approach to pharmaceutical development. So this is particularly ICH Q8 guideline, or more appropriately, we can say ICH Q8 R2 guidelines. It begins with predefined objectives. So objectives should be there in your mind. So now that I'm expecting the hardness of the tablets, this and this, dissolution, disintegration time. So this is the objective which you have set. It emphasizes product and process understanding and process control. The process and product understanding is most important. It is based on the sound science and quality risk management. Again, it is ICH Q9 guideline, that is the quality risk management. We'll see one by one. So here I have shown, so now there is the process understanding, product knowledge. And at the last I have shown it is a continual improvement process which is related to ICHQ10 guideline, that is the product quality system. So it is a continual improvement process. So it is not restricted only once you are developed and you will stop. So the knowledge every time which you will gain, so now that is you can utilize that for getting more quality into the product. So it should not stop. So process understanding, in that it is given the process design, the process performance, what are the processing parameters. So processing parameters, I will say, the what is the RPM of the blender, time for the mixing process. So that we can say that these are the processing parameters, and we need to keep the control on these parameters. Another thing is that it is given product quality attributes, control strategy, product specifications. So product specifications, desired product performance, it is given. So again, as I said, that is the limit which you have set for the quality, and whether you are getting that quality to the product. So that is the desired product performance. Process design. So process, the mixing process, what are the different parameters which comes, which need the control for the mixing process, granulation process. So all that. So now that is the control strategy is very important, and you should able to identify what are the critical parameters which can affect the quality of your product. So there are 10 steps involved in making the tablet, and if suppose for your tablet only 3 stages are the critical, you need to focus there only. So because you know that for 7 steps there is no risk involved, so for 3 steps you require more control. So what is the current approach? Current approach, if we'll talk, so this is by most of the pharmaceutical industries in India. They're working on quality by testing. So now there is a, for example, you are given the order for raw material, API, drug and excipient. You used to check that the raw material testing, then the manufacturing process होने के बाद उसका quality control करना, end products, uh, end product testing, then the fixed drug product manufacturing process. Because you applied to the FDA, you have taken the permission for the fixed dose and the fixed process, and accordingly you will manufacture. So now that is, you cannot make the changes unless and until you will inform to the FDA. So fixed drug product manufacturing process, end product testing. So now that is, this is fixed and thus inflexible process where we require the extensive testing. So it is a rigid process, what we can say in short, it is a rigid process, it is not a flexible process. So drawbacks, so we are testing few tablets out of several millions. It requires extensive in-process testing at every stage. We don't have permission to make changes in the operating parameters because these are fixed and approved by FDA. Any change if you need to make, we require the permission. We need to file supplements with the FDA and to get permission. So this is the drawbacks of the current system. So here if you see the ICH guidelines that make up the QBD family, these are ICH Q8R2 guidelines, that is the pharmaceutical development, ICH Q9 quality risk management, ICH Q10 pharmaceutical quality system. So these 3 parameters are important. So there are now up to ICHQ 14 parameters are given, but these 3 parameters are important. ICHQ8 guideline, that is related to the pharmaceutical product, ICHQ9 which is related to the risk management, Q10 which is related to pharmaceutical quality system. If you see for the ICHQ12, so this is for the technical and regulatory considerations for pharmaceutical product life cycle management. Some of the guidelines are related to the impurity profiling. So overall, it forms the QBD family. So, but most important 3 parameters, these are ICH Q8, Q9, and Q10 guidelines. So here, what was the need to have this QBD concept? So initially, अगर आपको product market में launch करना है, so we required to have the permission from the FDA. we require to submit the data to FDA. It may be related to the product, then the process, and the fixed dose. So now that is the fixed formula, sorry, rather than dose, it is a fixed formula we used to submit, fixed process we used to submit, and we used to get the permission for that. Now, as it is, I said that this is the continual improvement process. When you're working, you may find that we require to have some modifications. The quantity of the excipients we need to change slightly, or the mixing time, mixing speed I required to modify slightly. But the current process, it is a rigid process. It was a rigid process, and we required to take the permission, though we have to make any slight changes. And it happened in 2000. FDA, that is, we are talking about the US FDA, received approximately 5,000 supplements regarding these modifications in the formula, modifications in the process, and it becomes difficult for the FDA to go through these supplements and to give the permission. So this was the first incidence, and why there was a need to introduce this concept. To solve this issue in 2002, for the first time, FDA has introduced the concept of CGMP, current good manufacturing practice for the 21st century. In 2004, they published one final report, that is the pharmaceutical CGMP for the 21st century, a risk-based approach. And in that, they introduced the concept of quality by design. In 2005, the time came to implement this QBD approach. And from that, USFDA made mandatory. So for any product filing, manufacturers have to give the data as per QBD, quality by design guidelines. So what is this term quality by design, and who has introduced this concept? So for the first time, this concept was introduced by Joseph Juran in 1985. And he said, quality cannot be tested into the products. It has to be built by design. So quality accidentally नहीं मिलती है. We have to think, we have to plan, we have to design, and then only we can determine for the quality into the product. So I've shown here what is this quality by design. So it is the designing and developing of formulations and manufacturing process to ensure predefined quality. What it requires. So as I said that we require to have the understanding of formulation and manufacturing process variables and how they are affecting the quality of the product. What it can ensure. So it ensures for the product quality with effective control strategy. So what is this effective control strategy? If you're knowing, so these variables are going to affect the quality of your product. So we require to keep control on that parameters. We require to keep control on these parameters. So now that is, and if you keep the control on the variables parameters, we can ensure for the product quality. So QBD में क्या है expected? The product is designed to meet patient needs and performance requirement. It is very, very important. Patient is your customer, consumer. So quality is very important because he is paying from his pocket. The process is designed to consistently meet product quality, critical quality attributes. So quality attributes is different and critical quality attributes is different. We'll talk about that. The impact of starting raw materials and process on the product quality. The process is continually monitored, evaluated, and updated to allow for consistent quality over the period of time. So the process is continually monitored. So this is the continual improvement process. Critical sources of variability are identified and controlled. What are the possibility? What are the parameters, variables which can affect the quality of product? Identify and keep it control. Keep it in control. Science-based risk assessment is carried out. It offers a robust method or process. So method design concept helps to avoid cost involved. Initially, there was a resistance to introduce this concept, but later on, companies understood the significance of this quality by design. Initially, if you will invest, and if suppose the some experts will be available, so the post approval changes, the cost involved in the post approval changes can be minimized. So this is again the advantage. So it is an approach to product and process development and its consequences extending throughout the product life cycle. As I said, identify the variables which are critical to quality, identify the relationship between formulation and manufacturing process. So most of the time I have seen when the PG and PhD students are working, they are focusing mainly on the formulation aspect, the effect of excipients, but very rarely they used to focus on the processing parameters. But in industry, it is very, very important to focus on the both the parameters, that is the formulation and manufacturing process variables. That is substance, excipient attributes, process parameters, establish the relationship, that is the what is the effect of this formulation and process variables on the response, that is the quality of the product. This knowledge is used to implement a flexible, this word is very, very important, flexible and robust manufacturing process that can adapt and produce a consistent product over the period of time. So this process is a flexible process. How it is flexible, we'll talk on that. So where it is expected to have, so now that is container closure system. There also the concept of this QBD is important. You required to see the effect of this container closure on the stability of the product. So microbiological attributes are also involved. So these are the points from the QBD guidelines which are given. Next, we will see impurity profiling. So now that is the ICH Q3A, Q3B, and Q3C guidelines. So significance, if you see in US FDA, as per US FDA, initially we used to talk, our product is 99.7% pure, but now people are not interested in the purity. They're interested to know what is that 0.3% impurity. And as per US FDA, if you see in the US Pharmacopoeia, or the dossiers which are given. So in that it is mentioned, so which impurities are allowed and what is the limit of that impurities which are allowed in the product. If other impurities will come, so there might be possibility this will accelerate the degradation profile of the drug and stability of the product will be less. So for that, impurity profiling is important. So control on the impurities via QBD has benefits for patients, importantly for regulators, because if suppose my company is working, already innovator is there in US market and I am working on the ANDA and I want to file my product in the US market. So a regulator will ask first thing, what is the impurity, percentage impurity, which impurities are reported in your product, and if it not matches with the innovator or allowed limit, so your product will not get the permission into the US market. Now, QBT versus QBD approach to pharmaceutical development. As I said, quality is assured by testing and inspection of product in recent approach. In our country, still our FDA has not made it mandatory, but they are saying that that is if you submit the data along with QBD, that will be more appreciable. So there will be not much more data available during submission. Only testing data will be there. Specifications are based on the testing, that is on batch history. At a number of stages, we required to carry out the testing, and this become the complicated process. QBD approach. In QBD approach, if you see, quality is built into the product and process by design. Initially, क्या बनाना है आपको, प्रोडक्ट की क्वालिटी क्या होनी चाहिए, एट द एंड वी यूज्ड टू टेक, सो नाउ दैट इज लास्ट स्टेज क्या होनी चाहिए, आपका प्रोडक्ट का क्वालिटी क्या होना चाहिए, एंड फॉर दैट you have to think initially. So that is nothing but a designing. It is a knowledge rich submission. So now that is the product knowledge and process understanding. So for that, if you see, the process analytical technology PAT is given under that. Specifications based on product performance requirements. It is a flexible process within the design space. Design space concept क्या रहता है? So if you are expecting, if I'm saying dissolution of the tablet, so in 30 minutes, not less than 85%. इसका मतलब है 25 मिनट्स में अगर 80-90% आ गया, इट इज एक्सेप्टेबल. एनीथिंग अबव 85% इन लेस देन 30 मिनट इज द लिमिट सेट. बट अगर आपका टैबलेट एक घंटे में 70% हो रहा है, दैट विल नॉट बी एक्सेप्टेबल. सो डिजाइन आपका हो गया 80 to 100% इन लेस देन 30 minutes. So that is the flexible range which is given. If I talk disintegration time, so anywhere if you see, so now that is whenever we are talking for the percentage as say content uniformity, so the range is given. So we are working in the range. So the range is nothing but a flexible limit, and that is nothing but a design space. That is nothing but a design space, and you have to work within that space. Your knowledge space is there, inside that we can say the design space is there, inside that the control space is there. The control space is also important. So it focus on robustness, understanding and controlling variation. So benefits, it improves product and process understanding, it improves patient safety and efficiency, manufacturing efficiency, development efficiency. It helps for enhancing the process monitoring. It is continuous improvement process. It reduces end process testing, because in traditional approach we depends on the end result. So if you are keep the control on all the parameters, so there is no fear for the end product failure. So it reduce end process testing, it speed up the release decision. If suppose already you have supported your data with this QBD and you will send the next batch for the approval, so it will speed up. Facilitate change management. That is anything if you want to change, you not require the permission from FDA, because you are working within design space. Already for that design, FDA has given the permission. So now that is say for example, I want to say I'm using a granulator, the speed is within the this range to this range, time within this time to this time. Already I work on that. I've given that submitted the data to FDA, and FDA has accepted that data. Now, if suppose today you are keeping the speed RPM 20 RPM for 15 minutes, and next time you need to have 15 RPM for 15 minutes. So you need not to take permission from the FDA. Just you can drop one email to FDA, प्रोडक्ट जो लॉगिन रहता है आईडी, उसमें से आप उसको भेज दीजिए. So now these changes were made. Now instead of 20 rpm we are using 15 rpm for 15 minutes. So now that is you need not to take permission. But in traditional approach, once 20 rpm for 15 minutes time is fixed, if suppose 1 rpm change also you have to make, you need to have the permission. So facilitate the change management, fewer risk of failure, rapid response by FDA for any deviation in manufacturing, reduction of post-approval submissions, So now that is most of the time, if you see, we used to change. So post approval changes. So now that is, if you see the SUPAC guidelines, so there are less possibility for the post approval submissions. More efficient technology transfer to manufacturing. It is used to understand product specific requirement, and these are supported by the CGMP or GMP. What is the benefit to FDA and industry of employing QBD? It is very important. एफडीए में लोग कम है, number of submissions are there, regulatory submissions are there. But if suppose you will submit your product with QBD data, it enhances the scientific process for product review. Just they will go through the data, they will check the extreme limit which you have set, and results which you have got. The results are well acceptable. So now that is just only that they will go through that data, enhance the scientific process for product review. Better coordination between review, compliance, and inspection. More flexible. So range is given. So regulatory flexibility is given. Improving information in regulatory submission. Advantage to FD. For industry क्या हो जाता है देखिए, प्रॉब्लम ड्यूरिंग मैन्युफैक्चरिंग इज रिड्यूस बिकॉज ऑलरेडी आपने उस पर वर्कआउट किया है डाटा. So knowledge rich submissions is there, the knowledge rich submission to FDA. So data is gathered, it is available with industry. Reduce number of supplement during manufacturing. So there is no need because already your product has got the permission. It is helpful for understanding of risk occur during formulation and development. It is very, very important. You should be able to identify what is the risk associated. Risk means what? If suppose 10 excipient उसमें use कर रहे हैं हम and 3 excipients are very important. इसका quantity यहाँ वहाँ पे हो जाएगा, so that can affect disintegration time, dissolution, hardness, and all that parameters. So focus on that, and if you keep the control on that, so there is less possibility to have the risk failure into the product. It enables the continuous improvement in product and manufacturing process. So what are the key elements of QBD? So Q elements, if suppose you will see, these are the quality target product profile QTPP. So you define your product profile, number of parameters may be there. आपका टैबलेट का साइज इतना होना चाहिए, थिकनेस इतना होना चाहिए, कलर ये होना चाहिए, हार्डनेस. Then you can say content uniformity, assay value, DT, dissolution, many more. So these are the QTPP parameters. After that, critical quality attribute parameters. Out of that parameters, अगर कलर चेंज हो जाता है तो I least bother about that. Say for example, size थोड़ा बहुत यहाँ हो जाता है तो it is not a significant thing for me. But content uniformity change हो जाती है, assay value change हो रही है मेरी, DT change हो रहा है, dissolution change हो रहा है. Definitely this is the critical quality attribute for me. So identify that critical quality attribute parameters. Next thing is that risk assessment. Under the risk assessment, you can see there is a critical material attribute parameters and critical process parameters, CMA and CAPP. And it links to the critical quality attribute parameters. You use risk assessment tools. So the ranking is given. Say for example, अगर मैं माइक्रोक्रिस्टलाइन सेल्यूलोज में अगर फॉर्मुलेशन में यूज कर रहा हूँ टैबलेट बनाते टाइम, so that is not going to affect significantly to my content uniformity, say for example. But if suppose any drug is a BCS class 2, and if suppose I will use lactose as an excipient, that can affect the dissolution of the tablet. So you link these parameters CPP and CMA to the CQA parameters. Establish design space from knowledge space. The knowledge space is there inside that. As I said, knowledge space म्हणजे काय की आपल्याला माहिती आहे टॅबलेट बनवताना आपण MCC वापरतो, आपण लॅक्टोज वापरू शकतो डायल्युएंट म्हणून, सुपर डिसइंटिग्रेट वी कॅन यूज. सो दिस is known to us. So this is nothing but a knowledge space. From that knowledge space, in how much quantity generally it has to be used, you make the range, that is the design space. And inside that design space, control space is there, that is the control strategy. Design space, for that, design of experiment, modeling and multivariate statistical analysis. So now the next speaker may go in detail about this. Establish control space to ensure consistency of product quality. You can go for the process analytical technology. So I've seen in Cipla, Glenmark, so some of the companies, some experts are there who are working on this process analytical technology, quality response method, QRM. And as I said that the last process is the quality पे satisfaction होके रुकना नहीं है आपको. So once you have achieved this quality, you should not stop. You should go. That is the life cycle management, continual improvement beyond filing until discontinuation. So now that is what they said. So once you have filed, you should not stop improving the quality. You should continue unless you will withdraw the product from the market until discontinuation, it is said. Call it target product profile. So a set of elements that define the drug product. So there might be 10. The target or goal set in advance. A guide to drug product development is given. When to define QTPP? At the start of the product development. So knowledge gain in the development may change some elements. So that may change. अगर आप काम कर रहे हो तो काम करते करते आपको ध्यान में आ जाता है कि this is also one of the QTPP parameters. So now that is knowledge gain in the development may change some elements. Components related to safety, efficacy, identity, purity, and potency. So these 5 parts are very important. These are the parameters of the quality. Quality के parameters क्या बताया मैंने? So components related to safety, efficacy, identity, purity, and potency. Critical and non-critical components. So as I said that is the CQA. What is that critical? Critical में assay. अगर आपका assay value 80% आ रहा है, 80% assay value आ रही है आपकी, so it is not acceptable because the limit is given for the assay value 95 to 105%. So your tablet should be within the range of 95 to 105%. The assay. Content uniformity. और content uniformity बाद में बात करेंगे, उसमें भी limit है. So these are the critical parameters. But as I said that the appearance, some manufacturers have made it pink in color, some have made it some other color. So that is not significantly affecting. Fixed and variable components. So dosage form, we cannot change. The strength of the tablet, 250 milligram, that will remain forever. What are the variable elements? So for example, 250 milligram dose of the tablet, I'm making 450 milligram tablet. Some other manufacturer may make it 500 milligram, total weight of the tablet. So it is also not going to affect. Now, assay, again, it is important. So you should know what are the fixed elements, what are the variable elements. One example in short from the FDA guidelines, I've taken this example. So how it is QTPP is mentioned. So dosage form दिया है, target आपका रखा है, and this is for particularly the audience who is doing post graduation and PhD. So for that, it is important to have this target justification. So justification is important because FDA will not accept your data without justification. Another important thing is that those who are doing the PhD, तो प्री क्लिनिकल स्टडी अगर आप करते हो तो बहुत अच्छी बात है, बट सम ऑफ द projects I've seen without preclinical data, they have given in detail QBD data, so it is also acceptable. So dosage form modified release tablet है, फार्मास्यूटिकल इक्विवेलेंस रिक्वायरमेंट दिया है, सेम डोसेज फॉर्म, टैबलेट है तो टैबलेट ही होनी चाहिए, रूट ऑफ एडमिनिस्ट्रेशन ओरल है तो सेम रूट ऑफ एडमिनिस्ट्रेशन, डोसेज स्ट्रेंथ 10 मिलीग्राम तो सेम रहनी चाहिए। फार्माकाइनेटिक्स में टारगेट हमने दिया है कि फास्टिंग एंड फेड स्टेट कंडीशन में 90% कॉन्फिडेंस इंटरवल of the pharmacokinetic parameters. So after 2 hours, between 2 to 24 hours, and up to infinity time. And Cmax should fall within the bioequivalence limit. So this is the bioequivalence requirement which is given. So this is the target which you have fixed. So for me, the pharmacokinetic parameters will be important. So for me, the CQA may be related to the pharmacokinetic. Stability, this is also the CQA parameter. At least 24 months shelf life is important because the reference listed drug is reported to have the shelf life of 24 months. So reference listed, that is the innovator, drug product quality attributes, physical attributes, identification, assay, contain uniformity, degradation. So ये points है, इसमें से some of the points may be important for me. So these are the QTPP for the modified release product. It is given container closure system आपकी कौन सी होनी चाहिए. I given the justification. I will use the high density polyethylene bottles with child resistant cap and are selected based on the similarity with the RLD packaging. Innovator जो है उसका RLD packaging के हिसाब से मैंने बनाया है. So next, तो QBD for IR टैबलेट अगर मैं बात करता हूँ तो मैं उसमें क्या क्या आ जाएगा, डोसेज फॉर्म, तो मैं टैबलेट है तो टैबलेट से ही काम करूंगा, मैं टैबलेट का कंपैरिजन लिक्विड के साथ नहीं कर सकता. तो डोसेज फॉर्म इंपॉर्टेंट, डोसेज डिजाइन क्या है, रूट ऑफ एडमिनिस्ट्रेशन क्या है, डोज स्ट्रेंथ, कंटेनर क्लोजर सिस्टम, फार्माकाइनेटिक्स, ड्रग प्रोडक्ट क्वालिटी एट्रीब्यूट्स, it may be related to the tablet characteristics, it may be the hardness, dissolution, disintegration, friability. Then the identity, assay, contain uniformity, purity, impurity, dissolution, stability. So these are various number of parameters are there which we need to focus and fix. आप क्या एक्सपेक्ट करते हो उसमें, क्यों अभी इसमें से अगर आप जाते हो तो क्रिटिकल क्वालिटी एट्रीब्यूट नेक्स्ट आ जाता है आपका, कि मैंने जैसे बताया कि आपको 10 पैरामीटर आप डिसाइड कर सकते हैं कि क्यूटीपीपी में क्या होने चाहिए. आउट ऑफ दैट ऑल द पैरामीटर्स आर नॉट सिग्निफिकेंट फॉर मी and these are not affecting the quality of my product, but these three, say for example out of 10, 3 parameters are very very important. I need to keep control and you should be able to find out these 3 critical quality attribute parameters. So what is this CQA? It is a physical, chemical, biological, microbiological property that should be within an appropriate limit range to ensure the desired product quality. It may be physical, it may be chemical, microbiological, and that should be within the limit because these are going to affect the final product quality. So CQA is what? It is a subset of QTPP. It includes critical parameters that are likely to change based upon the variations in raw materials process. So identity test for dosage form, is it a CQA? I said that it is not a CQA parameter, but assay content uniformity is very important for me. I classified it as a CQA parameter. So CQA parameters need to monitor throughout a drug product development, and it required to ensure that that remains within the safe and effective levels. So critical process parameters and critical material attribute parameters, as I said, प्रोसेस में आपने वेइंग से लेके मिक्सिंग रहेगा, ग्रैनुलेशन रहेगा. मिक्सिंग में रहेंगे तो अलग अलग विच टाइप ऑफ द मिक्सर यू आर यूजिंग, व्हाट इज द साइज ऑफ दैट मिक्सर, व्हाट इज द स्पीड, for how much time you are doing the mixing process. All these are the processing parameters. Material attribute parameters में मैं MCC यूज कर रहा हूँ क्या, lactose super disintegrant यूज कर रहा हूँ क्या, what is the quality of that super disintegrant, which type of super disintegrant I'm using, what is the concentration of that super disintegrant. So these are the parameters which have a direct and significant influence on a quality, critical quality of my product. Process parameters include input operating parameters, mixing speed, flow rate, as I said, temperature, pressure, moisture. Material attribute parameters में मैंने बताया particle size distribution. So we rarely used to focus on this PSD, but it is very, very important. I've seen one of the case of A&A filing of particular product. Everything was same, process same था, excipient same था, but when they have gone for the pivotal bioequivalence study, pilot bioequivalence and pivotal bioequivalence study, they found that failure पहले तो आ रहा था अच्छे से, and what happened on of sudden when they have investigated, they found that the super disintegrant which they have used, उसकी particle size अलग थी. Initially the super disintegrant when they have used, and the final bioequivalence study when they have used, the polyplasodone XL and polyplasodone XL10. So what is that? How much difference it will make? The difference was of the particle size. So it is very important from company pharma point of view to focus on the particle size distribution, impurity profile, porosity, moisture level, and many more parameters. So the Q2PP to CQA. So QTP components, if you see, number of components I have mentioned. So dosage form, route of administration, strength, weight, pharmacokinetics, appearance, many more. So all that are related to the safety and efficacy. Safety and efficacy is important. Again, identity, potency, purity, toxicity. We are talking all these terms in related to the quality. So what are the potential quality attribute parameters? Out of the selected I have taken here, and then I've gone for the critical assessments. So because everywhere it is not possible to keep control, otherwise the study will become the extensive study and the filing will be late. So I've gone for the critical quality attribute parameters, and in that I found that the assay, it is affecting the efficacy of the product, impurities, it is affecting the safety, content uniformity, dissolution. So these four parameters are identified as a critical quality attribute parameters. So I need, so for that I said that the QTPP is important. After that, you should be able to find out the critical quality attribute parameters. So you focus only on these four parameters rather than other parameters. So risk assessment ICH-Q9. अगर अभी ये हो गया कि आपने आपका सीक्यू क्या है, क्रिटिकल क्वालिटी एट्रीब्यूट को आपने फाइंड आउट कर दिया कि ये चार पैरामीटर है, इट इज गोइंग टू अफेक्ट द एफिकेसी ऑफ माय प्रोडक्ट, सेफ्टी ऑफ माय प्रोडक्ट. So I will focus on that. Now you have to think, what are my processing parameters and the excipient parameters, material attribute parameters, which can affect this critical quality attribute parameters. Now, to study impact of each attribute which could have on drug product CQA, the relative risk that each attribute presents is ranked as high, medium or low. It is very important कि आपने अगर 10 एक्सिपिएंट यूज कर रहे, अभी आप लिखेंगे राइट हैंड साइड को ये आपका एसए वैल्यू, कंटेन यूनिफॉर्मिटी, डिसोलेशन एंड ऑल दैट. एंड व्हाट आर द मटेरियल एट्रीब्यूट पैरामीटर्स व्हिच विल अफेक्ट. सो अगर आप उसको लिखते हो कि ये पैरामीटर है, तो यू रैंक इट. Say for example, I'm using the lactose, whether the lactose is going to affect the content uniformity, whether the lactose is affecting the dissolution. If a BCS class 2 drug is there, I will say yes, it may affect at a medium level. If I will vary the concentration of the super disintegrant, whether it is going to affect the dissolution, I will say medium level for BCS class 2 drug. And whether it is going to affect the disintegration, yes, it is at the high level. So the relative risk that each attribute presents is ranked as high, medium, or low. The high risk attribute shows need for further investigation. How risky? How, this is the parameter is a risky one. Now in your class, if suppose some teachers are there, the class of 60 students is there. So now that is 5 students are there, which is affecting your class. So definitely there is a need to focus on that 5 students more. So these are at the high risk. Some are there, 10, 15 students are there. Okay, thoda it is affecting the class. Now you will classify it as a medium, and some are neutral. So उनका presence इतना class में problem create नहीं कर रहा है. So you will consider it as a low level. The high risk attribute shows need for further investigation. The low risk requires no further investigation. To assess the needs of additional studies for scale-up and technology transfer, identify appropriate specifications, critical process parameters, and manufacturing control. So these are the parameters. This is the all these guidelines are given on the USFDA website, and in this ICHQ 9 guideline, you will find this information. Risk assessment for formulation. अभी क्या है फॉर्मुलेशन में स्टार्टिंग मटेरियल प्रॉपर्टीज एंड लेवल ऑफ कंपोनेंट्स. जैसे कि मैंने बताया सुपर डिसइंटीग्रेंट इज अ स्टार्टिंग मटेरियल फॉर मी and what is the concentration of this super disintegrant in a fast disintegrating tablet. So this is the risk assessment parameter. मैन्युफैक्चरिंग प्रोसेस है, मैंने जैसे बताया था आपको कि मिक्सिंग है, ब्लेंडिंग है, ग्रैनुलेशन प्रोसेस है, कॉम्प्रेशन है. So whether it can affect the disintegration time of my tablet, so that you need to do the study on that. So steps for risk assessment: list out all components and processes, prepare the process flow chart, identify all potential failure modes for each atom with risk query, identify all potential failure modes for each atom and risk query. Risk query means what? So as I said, that high level, medium level, low level, risk query. Sometimes somewhere it is mentioned 1, 2, 3, 1 at low, 2 at medium, and 3 at high level. And you ask the question, justification. As I said, that justification is very important. What might go wrong? Do the risk analysis and then the risk evaluation. So this is important. So for ICH, so after ICH Q8, this is related to the product development, and this is ICH Q9 guideline, which is related to the risk assessment study. What are the methods or tools of risk assessment? So it is given failure mode effect analysis, failure mode effects and criticality analysis, fault tree analysis, so many more. The simple one, as I said, that is risk ranking and filtering, 1, 2, 3, or low, medium, high. Then some of the statistical tools are there you can make use of, because in industry people don't have time. They can afford to purchase softwares if it is available in the market, and they can process the data for the risk assessment. If you see, so it is given the risk priority number, what we have discussed for failure mode effect analysis. Ishikawa diagram, this is also there, that is the fishbone diagram analysis. It segregates risk into different categories. The risk which is associated with the machine, instrument, or the materials, the methods or process which are using, laboratory climate, the operators, that the human. So here I have shown, yes. So this is the Ishikawa diagram. So it is the Ishikawa diagram which I have shown. I said that is the blending inhomogeneity. So why this blending? So this is the fault tree analysis. You can see here first. Raw material properties क्या है, processing parameters, equipment properties. This can affect the blending inhomogeneity. I made raw material attributes. So I have written here raw material attributes. The processing parameters. So I have written processing parameters. Equipment properties. Then the environment, how it is going to affect. So this can affect my blending in homogeneity, because blend homogeneity is important for the content uniformity. अगर कंटेंट यूनिफॉर्मिटी नहीं रहेगी तो आपका ऐसे वैल्यू फेल हो सकता है. देन डोसेज यूनिट कंटेन नॉन यूनिफॉर्मिटी, दैट इज द टारगेट प्रोडक्ट प्रोफाइल. आपका क्या था कि आपका यूनिफॉर्मिटी होनी चाहिए थी. कंटेंट यूनिफॉर्मिटी वाज इंपॉर्टेंट फॉर यू. ऐसे इज डिपेंडेंट ऑन दैट कंटेंट यूनिफॉर्मिटी. तो ये आपका फेल जा रहा है. तो नाउ यू फाइंड आउट वेयर माइट बी द पॉसिबिलिटी समथिंग रॉन्ग. ना अभी क्या हो रहा है कि रॉ मटेरियल में आपने क्या किया है कि पार्टिकल साइज रेशियो है, पार्टिकल शेप क्या है, आपकी मटेरियल डेंसिटी क्या है, देन प्रोसेसिंग पैरामीटर्स में ब्लेंडिंग टाइम क्या रखा है आपने, आरपीएम क्या था. अगर मटेरियल अच्छा है, एवरीथिंग इज देयर एंड यू रिक्वायर्ड टू हैव द मिक्सिंग फॉर 15 मिनट्स एंड यू आर डूइंग जस्ट फॉर 5 मिनट, the speed which was expected was 20 and you are doing it at 10 rpm, so you will not get the blend homogeneity. Order of addition means what? The material quantity. Initially you are what adding? Excipient. You are adding the drug, or in between drug and excipient intermittent mixing you are doing. So that is important. Environment, what is the temperature and humidity. Equipment में blender का type क्या है, उसका volume क्या है. If suppose if we talk about the technology transfer, it is very important when you are going for the R&D scale up, R&D to pilot plant and pilot plant for to scale up. In that the technology transfer it is given. So total blender volume क्या रहता है, उसका बैपल का डिजाइन क्या है, blended. So all the parameters. So because of that the risk is associated. इसमें से कौन सा भी पैरामीटर आपके ब्लेंडिंग कंसिस्टेंसी को, होमोजिनिटी को कम कर सकता है. सो इंडस्ट्री पीपल विल नीड टू फोकस ऑन दैट. तो यही काम रहता है इंडस्ट्री वालों का कि प्रॉब्लम कहाँ पे आ सकता है, what may be the probable reason, so the assay value will be less or content uniformity will be less. So they will identify and will work on that. So this is known as the, you can see that this is just like a fish bone, and because of that this is known as the Ishikawa. So Japanese person was introduced this concept for the first time, and this is the fishbone diagram analysis. So this is the failure mode effect analysis. So this is the risk management tools what they used to do. So start quality risk management process. You identify the risk, go for the risk analysis, go for the risk evaluation. Risk आहे का आहे, काय होऊ शकतं, what can happen if suppose we'll increase the concentration or will increase the speed, will decrease the speed. और करके देखना है उसको. You go, you do the study and then the evaluate. That's the risk evaluation. So whether it is acceptable, accept कर रहे हैं तो process कर दीजिए. नहीं है, it is unacceptable. Again you focus on the risk analysis. I have shown here unacceptable है. Now रिस्क इवैल्यूएशन करने के बाद आपको ध्यान में आ रहा है कि हम अगर ऐसा काम करते तो आरपीएम ज्यादा रखते हो, 30 आरपीएम को मैंने रखा ब्लेंडिंग स्पीड, और उसको टाइम ज्यादा हो गया, तो देयर माइट बी पॉसिबली स्टैटिक चार्जेस में गेट डेवलप एंड द सेग्रीगेशन ऑफ द पाउडर बेड में ऑकर, एंड बिकॉज ऑफ दैट द एसए वैल्यू विल बी लेस. नाउ रिस्क रिडक्शन, हियर कंट्रोल आ जाता है, यहाँ पे आ जाता है कंट्रोल. अभी मुझे क्या करना चाहिए? सो आई विल गो फॉर द 10 आरपीएम फॉर 10 मिनट्स. I will go for the 15 rpm for 15 minutes, 20 rpm for 20 minutes, like that. I will go for the initial preliminary screening and I will gather the data, and then I will come to know I need to work within the range of 10 to 15 rpm and 10 to 15 minutes. Then only I can get the desired assay value contain uniformity. So my focus, my flexibility limit will be 10 to 15 rpm and 10 to 15 minutes. So now when I will submit the data to FDA, I will submit within that range. So this is nothing but the design space. So risk reduction, then the risk acceptance, result of quality response, and the review the events. So the process, as I said again, so this is continuous process. So now that is ऐसा नहीं है कि आज आपने काम किया और उसको देन देयर इज नो नीड टू इवैल्यूएट इट. So in between you can go for the evaluation and see, that is review the events. Risk assessment, QBD for ANDA. So very good example. Almost 200 pages document is given for immediate release, and 230 pages document is given for the modified release product on US web, FDA website. So QBD for ANDA example for immediate release. From there only I taken this example. Generic product development for acetylpyran tablets 20 milligrams. It is a BCS class 2 compound, so having poor aqueous solubility. It exists in three different polymorphic form, which may affect the dissolution. This is also the challenge for me. Polymorph 3 is the most stable polymorph, and drug prepared is with the roller compaction process, that is the direct compression process, or we can say the dry granulation blender in the dry form. Now the risk assessment. Now this is you required to give this data. Risk assessment for formulation components, drug products UKs. As I said that I have finalized the assay. There was 10 targets set by me, but out of that, four critical the targets I found that these are the critical quality attribute parameters. So I have fixed it to assay content uniformity, dissolution, degradation products, and formulation variables. So as I said that in industry, particle size distribution very important. Then I focus on the not only the MCC I use, but I vary the ratio of MCC in lactose. But as I said that as it is a BCS class 2 drug, and the lactose is a water soluble, so that ratio may affect the dissolution of my tablet. So blending करने के लिए MCC and lactose is important, and for that content uniformity मैंने उसको high level पे डाल दिया. Cross chromolose sodium level, so it is affecting the dissolution. Yes, definitely it is affecting the dissolution. Now if you can see this, what I said, the quantity of the magnesium stearate, whether it is affecting the dissolution, yes, it is at a high level. Degradation products, yes, sometime I found particularly with the aceclofenac. So magnesium stearate generally should not be used as a uh, lubricant. So because it can cause the problem for the stability. तो माइनर क्वांटिटी में डालते हैं अपन, मैग्नीशियम स्टीरेट डालेंगे तो लेस क्वांटिटी में डालते हैं, दैट आल्सो कैन अफेक्ट द स्टेबिलिटी. सो फॉर दैट आई हैव डन द स्टडी एंड आई हैव गिवन द रैंकिंग. इट मे हैपन व्हेन यू विल स्टार्ट विद द स्टडी, यू मे फाइंड कि ये इफेक्ट यहाँ पे सर ने दिया है हाई लेवल, फ्रॉम माय पॉइंट ऑफ व्यू इट विल बी एट द मीडियम लेवल. दैट इज योर परसेप्शन, but you need to give the justification for the same. As I say that 19-20 यहाँ वहाँ हो सकता है कि somewhere I may say that it is at a high level, you may say that it at the medium level. But it is not possible कि मैं हाई बोल रहा हूँ और आप लो बोल रहे हो, और आप लो बोल रहे हो मैं हाई बोल रहा हूँ. तो ये ऐसा हो जाता है कि सन इज राइजिंग फ्रॉम ईस्ट और आप बोल रहे हैं कि नहीं वेस्ट से हो रहा है, तो इट इज नॉट पॉसिबल. सो FDA विल नॉट एक्सेप्ट दैट डाटा. तो थोड़ा सा यहाँ वहाँ जो काम करता है, बिकॉज दिस इज द लॉजिकल थिंकिंग कि कहाँ पे प्रॉब्लम हो सकता है. जहां पे मैंने हाई बोल के बताया है कि ये एमसीसी लैक्टोज रेशियो हाई लेवल पे कंटेंट यूनिफॉर्मिटी को अफेक्ट कर सकता है. सम में से दैट सर इट इज गोइंग टू अफेक्ट एसे एट आल्सो मीडियम लेवल, इट मे हैपन. तो यहाँ पे आप मीडियम लेवल पे, तो जहां पे मैं हाई लेवल को बोल रहा हूं वहां पे कंट्रोल रखना मेरे लिए बहुत इंपॉर्टेंट चीज है. तो यहाँ की पार्टिकल साइज डिस्ट्रीब्यूशन इज वेरी इंपॉर्टेंट बिकॉज इट इज सिग्निफिकेंटली अफेक्टिंग द डिसॉल्यूशन. पार्टिकल साइज बड़े रहेंगे बीसीएस क्लास 2 ड्रग के. पॉलीमॉर्फिक फॉर्म स्टेबल पॉलीमॉर्फ बताया उन्होंने 3 नंबर का है, इसका मतलब उसकी सोल्युबिलिटी कम रहेगी. मुझे सोल्युबिलिटी बढ़ानी है, तो आई शुड नॉट यूज polymorph 3. So which polymorph I can use? At the same time, you have to think If suppose I will go for polymorph 1 and 2, whether it is going to affect the stability of my product, whether it is going to accelerate the degradation process of the drug formulation. तो ये सबका जो है, ये सिर्फ मैंने रिस्क असेसमेंट फॉर फॉर्मुलेशन कंपोनेंट्स बताया आपको। ये ड्रग मैन्युफैक्चरिंग जो है प्रोसेस का है, जिसमें प्री रोलर कॉम्पैक्शन, ब्लेंडिंग एंड लुब्रिकेशन। ये प्री रोलर कॉम्पैक्शन के पहले हम करते हैं ब्लेंडिंग। तो इसमें मैंने बताया कि द पैरामीटर्स आर केप्ट द सेम, ऐसे कंटेन यूनिफॉर्म डिसोल्यूशन डिग्रेडेशन। सो हाउ इट इज गोइंग टू अफेक्ट। नेक्स्ट स्टेज कम्स द रोलर कॉम्पैक्शन प्रोसेस, देन द मिलिंग। रोलर कॉम्पैक्शन करने के बाद आपने उसका जो बड़ा वाला स्लग बन जाता है उसको चॉप कर दिया, मिलिंग कर दिया। ये मिलिंग प्रोसेस अफेक्ट करती है कि मेरी ऐसे को। द फाइनल ब्लेंडिंग, lubrication, and the last stage is given the compression. So this is the dry granulation process. This is the dry granulation process. I have not gone for the wet granulation, but dry granulation. तो कॉम्प्रेशन करने के बाद बीसीएस क्लास 2 ड्रग को मैंने कॉम्प्रेस ज्यादा किया, उसका हार्डनेस ज्यादा रखा, तो डेफिनेटली इट इज गोइंग टू अफेक्ट द डिसोल्यूशन बिकॉज इट इज बीसीएस क्लास 2। बीसीएस क्लास 1 रहता तो इतना मुझे सोचने वाली बात नहीं थी, बिकॉज द ड्रग इज सोल्यूबल, जस्ट आई टू फोकस ऑन द डिसइंटीग्रेशन। बट हियर क्लास 2 के लिए मैंने बताया कि इसको हाई रखूंगा तो डिसोल्यूशन के लिए अफेक्ट करेगा। ब्लेंडिंग प्रॉपर नहीं रहेगी, क्वांटिटी ऑफ द लुब्रिकेंट इफ इट आई विल ऐड मोर, तो डेफिनेटली द डिसोल्यूशन विल गेट अफेक्टेड एट अ हाई लेवल। तो ये आपको बनाना बहुत जरूरी है। एंड देयर वी रिक्वायर टू गिव द जस्टिफिकेशन। ये सिर्फ अपन ओरली बोल रहे हैं, तो जहां पे आप हाई बोल रहे हो तो वहां पे आप हाई क्यों बोल रहे हो, सो जो टेबुलर फॉर्म में आपको देना है कि जस्टिफिकेशन आप क्या बता रहे हो उसको। सो डिजाइन स्पेस, इट इज वेरी इंपॉर्टेंट, बिकॉज अगर आप बोल रहे हो कि इसके लिए आप डिजाइन एक्सपर्ट सॉफ्टवेयर 14 नंबर है अभी, तो उसका ट्रायल एंड वर्जन आपको मिल जाता है फॉर 1 मंथ। ट्रायल वर्जन इज अवेलेबल। एक उसको आप अगर डाउनलोड कर लेते पीसी पे, तो वो आप उसको सीख सकते हो। मिनी टैप सॉफ्टवेयर इज देयर, stat is software is there, many more softwares are available, and you have to search which softwares are freely available. And if suppose your college can afford the cost, you can subscribe. So hardly I've seen for educational institute they are giving up to 30,000 rupees subscription. So design space, ICHQ और R2 guideline में क्या बताया उन्होंने, तो मल्टी डायमेंशनल कॉम्बिनेशंस ऑफ द प्रोडक्ट कैरेक्टरिस्टिक्स। अगर मैं बता रहा हूँ कि quantity of super disintegrant affecting DT and dissolution, this is one of the parameter. The ratio of MCC and lactose is affecting the dissolution, this is another parameter for me. At the same time processing parameter में मैं करता हूँ कि pre roller compaction affecting dissolution, final compression affecting dissolution। तो ये हो गए कि multivariate data, number of parameters is affecting the dissolution। तो ये अलग अलग डाटा हो गए, अलग अलग ग्राफ बन जाएंगे इसके। तो मुझे इंटरेस्ट है कि मेरी जो डिजायरेबल रेंज जो है, कि मैं एक्सपेक्ट करता हूं डिसोल्यूशन मेरा इतना होना चाहिए इतने टाइम में। तो इसके लिए मुझे एमसीसी लैक्टोज रेशियो क्या रखना पड़ेगा, सुपर डिसिंटिग्रेंट कॉन्सेंट्रेशन क्या रखना पड़ेगा। जो येलो रीजन में यहाँ पे आपको दिखा रहा हूँ, दिस इज माय डिजाइन स्पेस एरिया। तो ये रेंज है, ये जो है बिलो रेंज दिए और ये टॉप की रेंज दिए। तो इफ सपोज आई वांट my data should lie within this area, so here I have given that is the concentration। अगर अब मैं बताता हूँ कि स्टरिंग रेट है और दूसरा पैरामीटर है, तो उसको कितना रखना पड़ेगा, so that I can give। so 3-4 पैरामीटर्स को आप क्या करें, जो ग्राफ्स बनेंगे उसको आप ओवरले प्लॉट में डाल देते हो। ओवरले बोले तो क्या, एक के ऊपर दूसरे को आप फिट कर देते हो। एंड द एरिया कॉमन जो एरिया आ जाता है उसमें आपको लिमिट आ जाता है कि स्टरिंग स्पीड शुड बी दिस, आरपीएम शुड बी दिस, टाइम शुड बी दिस, क्वांटिटी ऑफ द एक्सिपिएंट शुड बी दिस। सो दिस इज इंपॉर्टेंट, ये डाटा आपको कलेक्ट होना इंपॉर्टेंट है। And this is the design space, this is the regulatory flexibility. I can work from this to this to this. So now I will say that this is the more appropriate design space for me, the area which is given in the yellow region. अगर आप सॉफ्टवेयर उसको यूज करते हो, डिजाइन एक्सपर्ट सॉफ्टवेयर इज कंपैरेटिवली इजी टू यूज, तो उसमें आप उसको और जो बच्चे पर्टिकुलरली एमफार्म के रहेंगे पीएचडी के, दे आर वेरी स्मार्ट, तो उनको सॉफ्टवेयर इजीली समझ में आ जाता है कि क्या करना है, जस्ट इट इज इनिशियली, और उसका एक्सप्लेनेशन दिया रहता है। आप सब अगर यूट्यूब पे जाते हो तो डिजाइन एक्सपर्ट सॉफ्टवेयर कैसा यूज करना है, अगर उसको देखते हो तो यू विल गेट द डिटेल इंफॉर्मेशन। उन्होंने बहुत बढ़िया उन्होंने उसमें ये किया है। सो मल्टी डायमेंशनल कॉम्बिनेशन ऑफ द प्रोडक्ट कैरेक्टरिस्टिक्स, इंट्रैक्शन ऑफ इनपुट वेरिएबल्स, देर इज, देखिए ह्यूमन ब्रेन व्हाट इट कैन डू, अगर एक वेरिएबल है इट इज अफेक्टिंग टू द क्वालिटी, वी कैन इंटरप्रेट, इसको ज्यादा किया तो क्या होगा, कम किया तो क्या हो जाएगा, मैक्सिमम क्या कर सकते हैं आप, ए एंड बी, दिस टू वेरिएबल्स आर देयर, हाउ इट इज अफेक्टिंग द क्वालिटी। बट if I say A, B, C, D, चार वेरिएबल्स आर देयर एंड थ्री रिस्पॉन्सेस आर देयर, तो ये कैसे अफेक्ट कर सकते हैं, तो इसको एनालाइज ह्यूमन ब्रेन से करना समवॉट इट इज डिफिकल्ट। सो रिग्रेशन एनालिसिस का जो डाटा देते हैं, सो मल्टीवेरिएट रिग्रेशन एनालिसिस, अगर आप स्टैटिस्टिक्स का बोल्टन का बुक देखते हो, स्टैनफोर्ड बोल्टन का, तो उसमें वो दिया है उन्होंने। अगर आप ऑनलाइन भी सेशन देखते हो तो उसमें दिया है। So interactions of process parameters you have to check. Changes within the design space are not considered regulatory change. It is very, very important statement. What FDA is saying, any change which you will do in the design space is not a regulatory change. इसका मतलब है कि अगर आप कुछ चेंज करते हो और वो डाटा अगर आप एफडीए को पहले अपने स्टडी करके भेज दिया है, तो एनीथिंग, so this is the playground, I will say that you can keep your variables fixed here or here or here, you need not to take permission again from the FDA for making any change. QBD information and conclusions need to be shared with the FDA. From FDA perspective, regulatory submission in regards to design space should include description of design space, critical and other relevant parameters. Design space can be presented as the ranges of material input process parameters, graphical pre-present, contour plot है, इसको भी कैसे इंटरप्रेट किया जाता है by graphical presentation, वो भी दिया है, because within one lecture it is not possible to discuss on all these aspects. Contour plot, response surface plot, interaction, overlay plots क्या रहते हैं, then the mathematical relationships. So I will say the relationships may be linear relation. Say for example, simple अगर हम करते 3:2 factorial design के बारे में अगर बात करेंगे, two variables are there, that is the concentration of the lactose and the concentration of the super disintegrant, which is affecting the disintegration time and the percentage drug release. Simple case study जो हमने 2011 में हमने फर्स्ट टाइम मेरे एमफार्म स्टूडेंट के साथ किया था हमने। तो इसमें क्या हो गया कि इट इज अ सिंपल रिलेशनशिप, अगर मैं लैक्टोज का कॉन्सेंट्रेशन बढ़ाता हूँ और सुपरडिसिन क्वांटिटी बढ़ाता हूँ, सो इट इज पॉजिटिवली अफेक्टिंग द डिसइंटीग्रेशन, और नेगेटिवली, सो नो दैट इज कॉन्सेंट्रेशन बढ़ाने के बाद डिसइंटीग्रेशन टाइम कम हो जाएगा, सो इट इज नेगेटिवली अफेक्टिंग। तो इट इज अ लीनियर इक्वेशन। सो लीनियर इक्वेशन मीन्स क्या है कि परसेंटेज dt is equal to some fixed variable is there plus a plus b. So now the a plus b जो है आ जाता है तो it is a linear, अगर उसके साथ plus a b आ जाता है, that is the interaction variable is there. interaction variable, a का individual effect है, b का individual effect है, and along with that ab का भी effect है, so that is the interaction variable. So that relationship is known as the 2f1 relationship. Sometime it may happen कि वेरिएबल्स ऐसा भी नहीं रहता कि हमेशा कि आप वेरिएबल्स की क्वांटिटी बढ़ा दो तो dt कम हो जाता है, और वेरिएबल की क्वांटिटी बढ़ा कम कर दो तो dt बढ़ता है, अप टू सर्टेन लिमिट इट में हैपन कि हो जाता है, और फिर अगेन रिवर्स हो जाता है उसका. तो it is not a linear relationship, so it is known as the quadratic relationship. And whenever the quadratic word will come, so it is a plus b plus ab plus s square plus b square plus s square b square. Still more complicated mathematical relationship is there, the cubic relationship is there. तो इसलिए मैं बोल रहा हूँ कि सॉफ्टवेयर इज नीडेड फॉर दैट पर्पस. ह्यूमन ब्रेन कैन नॉट डू ऑल दिस प्रोसेसिंग. अभी आप बोलोगे कि ये हम डीओई करते और डिजाइन ऑफ एक्सपेरिमेंट करते हो, किस लिए करना है? तो प्रिलिमिनरी स्क्रीनिंग करना है आपको. प्रिलिमिनरी स्क्रीनिंग में वेरिएबल्स को सिलेक्ट करने रहते हैं, एंड जो मोस्ट इंपॉर्टेंट वेरिएबल्स है उसको आप फैक्टोरियल डिजाइन में लगा दीजिए. 3:2 फैक्टोरियल डिजाइन में जाइए, और आप बोलते हैं बॉक्स बेंकन डिजाइन. तो प्रिलिमिनरी स्क्रीनिंग डिजाइन है, दैट इज अ प्लैकेट बर्मन डिजाइन बोलते हैं उसको. बाकी जो डिजाइन है आपके, तो सिंपल वाला 3:2 है, दैट इज नोन एज द फुल फैक्टोरियल डिजाइन. नेक्स्ट डिजाइन आप बोलोगे तो बॉक्स बेंकन है, सेंट्रल कंपोजिट है, सिंप्लेक्स. अगर आप सॉल्वेंट मिक्सचर यूज कर रहे हो और उसके सोल्युबिलिटी पे इफेक्ट देख रहे हो, तो व्हेनेवर द कंसेप्ट ऑफ मिक्सचर विल कम, यू विल यूज द सिंपलेक्स डिजाइन, simple mixture design we can say. So various designs are there, it is given. तो उस कौन सा डिजाइन यूज करना है, इसके लिए भी आप यूट्यूब पे अगर देखते हो तो यू कैन गेट इंफॉर्मेशन. अगर कोविड का कुछ फायदा हुआ है तो आई विल से दैट कि आप इंटरनेट पे ज्यादा बैठ के उसको सर्च करना सीख गए आप. सो यू कैन गेट इंफॉर्मेशन फ्रॉम इंटरनेट, यूट्यूब. So as I said that is the design space, material, process, and how it is affecting the product. So material and process के बारे में अगर आप देखते हो तो parameters are important, which is affecting the material, which is affecting the process, and this is quality affecting to the quality of the product. So control space, as I said, use the word. This is the unexplored space. Layman, he don't know anything. लैक्टोज क्या रहता है, एमसीस क्या रहता है, उसको कुछ लेना देना ही नहीं है. Hence I said that it is an unexplored space. If I'm not expert in the nanoparticulate drug delivery system, so it is totally unaware of me, or just I'm having the knowledge. If suppose I have to make the biodegradable nanospheres, तो उसमें क्या यूज करना है, some knowledge is having, that is the knowledge space. Then the design space is there, and in that design the control space, that is the target or normal operating ranges. इसमें आप काम करते हो तो your batch will not fail. So ये एक डिजाइन स्पेस का कंसेप्ट है. Designing and implementing control strategy ensure that material and process is within the expected lower and upper limits. Limit set किया आपने. Parameter and material are routinely controlled during production in order to assure reproducibility of the product. The control space should be within the design space. Scale up is trial and error basis. During scale up process, parameters may differ, but attributes which affect quality remain the same. And hence control strategy is required. Control strategy. One case study is there. उसमें बताता हूँ मैं. Control strategy includes input material attributes related to the drug substance, excipients, container closure, equipment operating conditions, that is the process parameters. Some controls are in process controls, which is affecting the finished product quality. Control for each unit operations, it is important. मैंने जैसे बताया कि आरपीएम क्या होना चाहिए, आपका मिक्सर क्या होना चाहिए, उसकी स्पीड क्या होनी चाहिए, टाइम क्या होना चाहिए, वॉल्यूम क्या होना चाहिए. तो कंट्रोल ऑन ईच यूनिट ऑपरेशंस. So life cycle management, continuous improvement help to identify and implement appropriate product quality improvement. Process improvement, so product quality improvement, process improvement, reduction in the variability. It leads to innovations and pharmaceutical quality system enhancement, but for continuous improvement and process improvement, it should be within the original design space. जो कुछ काम करोगे आप प्रोडक्ट का पहले फाइलिंग कर चुके हो, क्यूबीडी का स्टडी कर चुके हो, रेगुलेटरी फ्लेक्सिबिलिटी आपको अप्रूवल मिल चुका है, बट दिस इज विद इन द डिजाइन स्पेस. अगर कुछ भी आपको करना है, सो यू नीड टू वर्क विद इन दैट डिजाइन स्पेस ओनली. So tools of QBD, DOE. So according to FDA guidance under Q8 R2 guideline related to the pharmaceutical development. So design of experiments क्या रहता है? A structured, organized method for determining the relationship between factors which affect a process and the output of that process. इसका मतलब है कि number of parameters which affect the quality of the product. You find out the relationship between that, and generally this is the mathematical relationship. आपको एक इक्वेशन मिल जाता है, और उसमें एक इक्वेशन जो मिल जाता है आपको, तो वो इक्वेशन में आप थोड़ा सा चेंजेस उसमें क्या करते हैं, फिक्स वेरिएबल्स रहते हैं, क्वांटिटी ऑफ ए बी एंड सी यू कैन चेंज, एंड द इक्वेशन. तो मिनिमम नंबर ऑफ एक्सपेरिमेंट्स कैन गिव यू द extensive data. It is a structural plan for conducting an experiment, measure the effect of input on output variables, determine which factor is critical to the response, it is cost effective, it helps in gaining maximum information from minimum number of experiments. 9 experiment कर दीजिए, equation मिल जाएगा. Now just you change the value of A and B, accordingly you will get the value of the response, because with the 9 experiments only you have got the mathematical equation. Why and when to conduct DOE? Evaluate the effect of formulation and process factors on critical quality attribute parameters. Confirm critical material attributes, formulation factors, processing parameters. Identify interacting variables. As I said that only linear relationship. So A is affecting to the response, B is affecting differently, then A plus B affecting, and plus AB. So now that is the interaction variables. Optimize the formulation, develop the design space. So DOE models, it is given placket Berman design. So it is carried out at the early stages for screening. This is the preliminary screening model. So possible to neglect higher order interactions. It is 2-level multi-factor experiments. अगर 11 वेरिएबल्स है तो 12 एक्सपेरिमेंट्स ओनली करने पड़ते हैं आपको. 11 वेरिएबल्स है, आपके 99 वेरिएबल्स है, 100 एक्सपेरिमेंट्स करो, आपको उसमें से समझ में आ जाता है कि which variables are important for you. When number of factors involved is higher, to economically detect large benefits. And as I said that it is possible to study 7 factors in 8 runs, 11 factors in 12, and 99 factors in 100 runs. So this is the placket Berman design. If you go in design expert 14 version, so now that is, you will find that it is given placket Berman design. Box Benken. So the design requires fewer runs. So if suppose the number of variables are 3, if suppose you will go by the 3:3 full factorial design method, you need to go for the 27 experiments. But I want in less number of experiments, the data, I can go for the Box-Benken design. Design requires fewer runs, 15 runs in a 3-factor experimental design. The software gives 15 run design, where the 12 combinations of the factors and 3 the center point. जो अलग अलग पॉइंट्स है वो 12 पॉइंट्स बन जाते हैं. उसको जो अगर आप देखते हो एक्सप्लेनेशन, तो स्क्वायर दिखाए, क्यूब दिखाए, और क्यूब के अलग अलग पॉइंट्स पे नंबर ऑफ अगर आप देखते हो तो 12 एक्सपेरिमेंट हो जाते हैं. एंड द सेंटर पॉइंट, सो दिस, और अभी ये प्रॉब्लम बच्चों को ध्यान में नहीं आती है. दे आर सेइंग दैट ये 3 जो पॉइंट आ रहे, दीज आर द रिप्रोड्यूसिबल पॉइंट, रिपीटेड पॉइंट्स है. तो एक बार ही बच्चे करते हैं, और उसका वैल्यू वैसे का वैसा रख देते. It is not like that. Though the value is same for three combinations, you required to do the study in the triplicate. और उसका अलग अलग रिजल्ट आ गया तो आप अलग अलग रिजल्ट को उसको मेंशन करना है. These are the center points to estimate the pure error. When it is required to economically detect large mean effects, when it is expensive to perform all the necessary runs. So now the 27 runs करने से better है just you go for 15 runs and get the data. Factorial design, full factorial, when the number of factors and levels are small, it is the most preferred design. As I said that 2 raised to 3. What do you mean by 2 and 3? So 3 variables at 2 level of study. If when I'm using the word 3 raised to 2, 2 variables at 3 level of the study, my 2 variables, say for example, lactose and the super disintegrant. So these are the 2 variables. What is this 3 level of study? I used to write +1, 0 and -1. What is this +1, 0 and -1? So I will say that the lactose I will use 40 milligram, or I can use in the range of 20 to 60 milligram. So 20, 40 and 60. So 0 value will come to 40, +1 value will be 60, and -1 will be 20. So one experimental unit stands for 20 milligram of lactose. Simple disintegrant concentration, if I'm using 5, 10, 15. So for 0, it may be 10, -1, 5, and +1, 15. So full factorial design, when we want all possible interaction information. Fractional factorial design is there. Central composite design, it is again the each factor has 5 levels. That is here, if you see, it is given -1, minus alpha, 0, plus alpha, and +1. Hence, it is said this factor has 5 levels. A central composite face centered response study. So now that is these are 13 trials, 08 trials with 2 center point replicates and 3 non-center point replicates. So 32 trials. So alpha and minus alpha value. So the software itself will calculate and will tell you what is this alpha and minus alpha value. Simple mixture design, as I said, the blend of alcohol and water, and you are doing the study of solubility. So the best design is to go for the simple mixture design. The softwares if you use, so here I should mention 13, but latest it is 14 design expert software and many tabs. That is, so number of softwares are there. Half normal probability plot, you may have seen that. NOI is given, half normal probability plot is given. So ये अगर आप plot करते हो, predicted R square, actual R square, that is the observed R square, उसको देखा जाता है. P value is given, तो P value is less than 0.05. So the probability hypothesis पे depend करता है, आपका hypothesis क्या है, और उसके हिसाब से P value आप क्या बोल रहे हो. तो P value is less than 0.05. So the study, the factors which you have selected, the level of that factors which you have selected, it is significant, and these are significantly affecting your study. That is the significance of the p-value. So model terms A and B, these are significantly affecting to the study. So half normal probability plot used to distinguish between important and unimportant effects. Number of variables are there, A, B, C, if it is there. And say for example, you can say A का B के साथ interaction हो सकता है, A का C के साथ हो सकता है. Then you can say the interaction variable is A, B, C. So which one is important and which one is unimportant? So primary grouping of factors and interactions. Secondary ranked list of factors and interactions from most important down to least important. तो उसको रीड कैसे किया जाता है, देखिए वर्टिकल एक्सिस दिया है लार्जेस्ट टू स्मॉलेस्ट, लार्जेस्ट टू स्मॉलेस्ट तक दिया है. So absolute values of the estimated effects of the main factors and available interactions. The line, this line, ये सबसे important जानने वाली बात है. The line indicates the values when all factors are at a zero level. The points away from line towards the right, these are the points. So these points which you can see, दिस इंडिकेट्स दिस इज द सिग्निफिकेंट इफेक्ट। तो इसका जो बोलना है कि ये लाइन का है वो जीरो वैल्यू की लाइन है। तो यहाँ पे अगर आप एक्सीपीएम को अगर रखते हो तो दिस इज नॉट अफेक्टिंग टू द स्टडी, सो दिस ऑल फैक्टर्स आर एट अ जीरो लेवल। अभी अवे फ्रॉम हो गया, द पॉइंट्स अवे फ्रॉम दिस लाइन टुवर्ड्स राइट इंडिकेट सिग्निफिकेंट इफेक्ट। The point extreme right is significantly contributing to the study. You can see the A is the point at its extreme right. AC is also the point. Away from this line, jitna away from line रहेगा towards the right hand side, it is significantly contributing to the study. तो ये जानना बहुत इंपॉर्टेंट। बच्चों को ऑब्जर्व प्रिडिक्टेड रिस्पॉन्स क्या है, उसका आर स्क्वायर क्या है, आपका प्रिडिक्टेड रिस्पॉन्स क्या है, प्रिडिक्टेड आर स्क्वायर वैल्यू क्या है, आपके ऑब्जर्व कितनी आ गई। सो एनोवा टेबल दिया है, एनोवा में क्या बताया, पी वैल्यू क्या बता रहा है, तो इसको रीड करना इंपॉर्टेंट है। इक्वेशन दिया है y इज इक्वल टू एक फिक्स वेरिएबल, अगर आप ऐसा पकड़ के चलो कि y जो है परसेंटेज ड्रग रिलीज इज इक्वल टू 30 प्लस 2.83 ए माइनस 2.57 बी। तो मैंने जो बताया कि ये वैल्यू जो है उसको मैं बोल रहा हूँ कि कोएफिशिएंट जो है these are fixed values. What is going to change? Only the value of a and b is going to change. अगर मैं प्लस a बोल रहा हूं तो इसका मतलब as I will go on increasing the concentration of a, it is positively affecting the percentage drug release. a को मैं बढ़ाता हूं तो मेरा परसेंटेज ड्रग रिलीज भी बढ़ने वाला है। इफ आई एम यूजिंग माइनस b वैल्यू, minus sign indicates that if I will increase the b, it is going to negatively affect the percentage drug release. तो ये इंपॉर्टेंट है कि इक्वेशन में क्या दिया जाता है। सरफेस जो है रिस्पॉन्स सरफेस पैथोलॉजी, आरएसएम बोलते हैं उसको, ये रीड करना आपको जरूरत है। कंटूर प्लॉट दिया है, कंटूर का मतलब भी ऐसा है कि बाबा जैसे आपने वेदर फोरकास्टिंग देखते हो कि ये एरिया में संभावना है बारिश की, और जो है हाई प्रेशर बेल्ट इज देयर, लो प्रेशर बेल्ट इज देयर। सो ये ओनली पर्टिकुलर गांव के रिलेटेड नहीं, एक जोन आ जाता है, जोन एरिया। तो ये इसमें ये होने की संभावना है। एंड एट द सेंटर पॉइंट्स यू विल फाइंड दैट इट इज मोर अफेक्टिंग, एंड इफ यू गो अवे फ्रॉम दिस इट इज नॉट सिग्निफिकेंटली अफेक्टिंग। यहाँ पे अगर आप देखते हो कि मैंने दिखाया था, ये जो है वो मेरा इसका है HPMC K100M एंड पॉलीऑक्स WSR 301। तो ये हमने यूज किया था कि कॉम्बिनेशन ऑफ टू पॉलीमर्स फॉर सस्टेन्ड रिलीज इफेक्ट, एंड आई सीन द रिलीज ऑफ द ड्रग आफ्टर 12 आवर्स। तो 12 आवर्स के बाद उसका रिलीज क्या होता है। सो व्हेन आई हैव ये प्लस वन वैल्यू ये जगह पे आ गई मेरी, प्लस वन एंड माइनस वन यहाँ पे आ गई, तो मेरा कॉन्सेंट्रेशन अगर HPMC और पॉलीऑक्स का ज्यादा रहेगा तो परसेंटेज ड्रग विल मिनिमम, और अगर मेरा कम रहेगा, यहाँ पे अगर आप देखते हो कम रहेगा यहाँ पे, तो द परसेंटेज ड्रग रिलीज, सिंपल बात है 10% HPMC मैं यूज कर रहा हूँ प्लस 10% पॉलीऑक्स, और मैं 5% प्लस 5% यूज कर रहा हूँ, 7.5 प्लस 7.5। तो आई फाउंड दैट 10 प्लस 10 गिव्स लेस रिलीज टू मी। तो यहाँ पे मुझे अगर आ जाता है तो यहाँ पे मुझे इक्वेशन भी आ जाता है, इक्वेशन। तो दैट इक्वेशन इज इंपॉर्टेंट। Next speaker will discuss, as I said, in detail about this Pareto chart analysis. So what is the Pareto principle? It is referred as the 80-20 rule. इसका मतलब क्या है देखिए, एक पर्टिकुलर ऑर्गेनाइजेशन है, ऑर्गेनाइजेशन 100 पीपल की है, 100 people are working, so the 20 people are contributing to the 80% profit of that organization. इसका मतलब this 20% people are important people for the organization, and 80% of the people are affecting to the 20% profit of the organization. So this is known as the 80-20 rule. So now that is, you can see here it is given, so ECA. So now that is, if you see, so this is the significantly affecting to the study, the E variable is affecting. A Pareto chart is a series of bars whose heights reflect the frequency or impact of problems. Bars on left are relatively more important. So these are toward the left hand side are relatively more important than the bars on the right hand side. You can see the height uski kam hoti ja rahi hai. Helps in identifying most critical problem to solve as well as the level of criticality. तो ये पैरेटो चार्ट भी बहुत इंपॉर्टेंट है. हाफ नॉर्मल प्रोबेबिलिटी इज इंपॉर्टेंट. ये प्रोसेस एनालिटिकल टेक्नोलॉजी दी है कि जैसे मैंने बताया कि अभी क्वालिटी बाय डिजाइन है. एवरी टाइम टेस्टिंग इज नॉट पॉसिबल. तो आप इनलाइन, एटलाइन एंड ऑनलाइन प्रोसेस रहते हैं जिसमें आप प्रोसेस को मॉनिटर कर सकते हो. से फॉर एग्जांपल मैं ब्लेंड यूनिफॉर्मिटी टेस्ट करना चाहता हूँ. I should not stop the process and take the sample by quality control department. I can insert vertical probe. The process is going on and it is connected with the software. I can monitor by sitting here कि उसकी स्टेज क्या है, कि अभी जो मैं मिक्सिंग कर रहा हूँ तो प्रॉपर मिक्सिंग उसका हो रहा है क्या, अभी ये इतना टाइम में मिक्सिंग मेरा रहेगा, मिक्सिंग इफिशिएंसी सो एंड सो. तो सम प्रोसेस एनालिटिकल टेक्नोलॉजीज आर देयर. समटाइम यू रिक्वायर्ड टू डाइवर्ट. अगर मेरा रिएक्शन चल रहा है, रिएक्शन केमिकल रिएक्शन चल रहा है, तो आई शुड नॉट स्टॉप द केमिकल रिएक्शन, देन इट टेक द सैंपल, गो फॉर द एफटीआर स्टडी, देन उसका रिजल्ट आएगा फिर मैं चालू कर दूंगा. नो, सैंपल डाइवर्ट करना है, दैट विल गो टू द एफटीआर. प्रोसेस आपको चालू रखना है. सो नो दैट इज द डाटा यू विल गेट. स्टिल इट रिक्वायर्स द रिएक्शन इज नॉट कंप्लीटेड. तो ये जो है, सो टेक्निक्स यूज दीज आर गिवन. Raman spectroscopy, near IR spectroscopy, NMR, FTIR with ATR probe. So these techniques are used in the granulation, compression, crystallization, mixing, endpoint detection. तो ये भी सपोर्ट करता है, पैट जो आपका है वो सपोर्ट करता है क्यूबीडी को. अगर पैट का डाटा भी अगर आप देते हो एफडीए को, तो आपको ज्यादा इंफॉर्मेशन एफडीए के लोग पूछते नहीं है. So these are the PAT tools as a given. So in conclusion, I will say QBD has gained importance in the area of pharmaceutical processes like drug development, formulations, analytical method, and biopharmaceuticals. As I said, it is not being a pharmaceutics person. मैं आपको formulation development के बारे में बता रहा हूँ. It may happen some analytical person may be there, तो वो analysis के बारे में बताएगा. HPLC पे काम कर रहा है, तो HPLC पे काम करते टाइम उनको area under curve is important. फिर solvent का mixture जो लेंगे वो, so now that is उसका जो पंप का प्रेशर रहता है, उसका कॉलम कौन सा यूज करते हैं. तो दीज आर द वेरिएबल्स एंड विच इज अफेक्टिंग द रिस्पॉन्स. पिक रिस्पॉन्स क्या रहेगा, एयूसी क्या रहेगा, टेलिंग आपको कम करनी है उसकी. तो ये जो पैरामीटर आ जाएगा वो एनालिटिकल वाला बात करेगा. तो इसका मतलब एनालिटिकल में भी क्यूबीडी का सिग्निफिकेंस है. अगर ऐसा नहीं कि फार्मास्यूटिक्स वाले बच्चे ही क्यूबीडी करेंगे. बायोटेक का फर्मेंटेशन आप करते हो तो उसके रिलेटेड अगर आप कुछ है, उसमें जो प्रोसेसिंग पैरामीटर्स रहेंगे, पीएच रहेगा, तो उसमें भी आपको कंट्रोल करना है. दैट इज अफेक्टिंग द क्वालिटी, यील्ड वैल्यू. सिंथेटिक का आदमी है तो उसमें भी इंपॉर्टेंट है. तो इट इज गिवन. So QBD has gained importance in the area of pharmaceutical process like drug development, formulations, analytical methods, synthetic, biopharmaceuticals, biotechnological product. Not restricted to pharma only, but in engineering mostly this QBD approach is also used. The main reason behind the adoption of QBD in the regulatory requirement is the regulatory requirement. As I said, अगर फिक्स एंड रिजिड प्रोसेस रहेगा तो आपको क्या करना है, आपको परमिशन लेनी है कि कुछ चेंज कर रहे हो आप, तो मैं यहाँ पे थोड़ा सा चेंज कर रहा हूँ. सेंड मेल, सेंड सप्लीमेंट, फी भर दीजिए उसकी, और फिर फी तो आप भर देंगे, 5,000 रुपए रहेंगी और कुछ रहेगी तो उसको फी कंपनी पे कर देंगे. बट इट बिकम्स हेक्टिक जॉब फॉर एफडीए. नंबर ऑफ सप्लीमेंट्स नॉट फॉर न्यू प्रोडक्ट बट ओनली फॉर द मॉडिफिकेशन. सो नाउ देयर इज स्लाइट वेरिएशन. तो इससे अच्छा है कि आप क्या कर दीजिए, पहले ही रेंज में परमिशन ले लीजिए कि हम इसमें इतना तो इतना क्वांटिटी में काम करेंगे, इतने टू दिस रेंज में हम प्रोसेसिंग पैरामीटर्स हमारा रहेगा. और अगर इसको अगर आपको परमिशन मिल गई, तो एनीथिंग इफ यू डू विद इन दैट डिजाइन स्पेस, so now that is it is acceptable. So pharmaceutical industry needs a regulatory compliance so as to get their product approved for marketing. ये अगर डाटा देते हो तो फार्मास्यूटिकल इंडस्ट्री को परमिशन मिलती है, कंप्लायंस जो पोस्ट कंप्लायंस रहता है प्रॉब्लम वो कम हो जाता है. क्यूबीडी रिप्लेसेस प्रीवियसली यूज फ्रीज अप्रोच ऑफ प्रोसेस डेवलप. फ्रीज बोले तो फिक्स. बाय प्रोवाइडिंग अ डिजाइन स्पेस कंसेप्ट, moving within design space would not require post approval changes. मूविंग विद इन डिजाइन स्पेस, एज आई सेड दैट, यहाँ से वहां और वहां से यहाँ डिजाइन स्पेस में अगर आप काम कर रहे हो तो आपको पोस्ट अप्रूवल चेंजेस की जरूरत नहीं है. इट रिड्यूसेस द कॉस्ट इन्वॉल्व देयर विद. यूएसएफडीए हैज मैंडेटेड क्यूबीडी अप्रोच टू जेनेरिक ड्रग प्रोडक्ट्स फ्रॉम जनवरी 2013. कोई भी प्रोडक्ट आपको अगर एएनडी के लिए फाइलिंग करके बाकी कंट्री से यूएस में जा रहा है तो इट इज मैंडेटरी to submit a QBD data. Recently, I talked with one of my student who is working in CDSCO, New Delhi. They said that sir हम भी उस बोलते हैं कि आप QBD से we are not made it mandatory, पर हम उनको encourage करते हैं कि ये डाटा दे दीजिए in this format. It is ultimately helpful to regulatory bodies for inspection and review process, but it will avoid loss raised due to hurry and unethical struggle of private firms to market their product as early as possible. That is very important because there is a brutal competition. कौन सी कंपनी फर्स्ट फाइलिंग करते हैं तो उसको फर्स्ट क्लेम मिल जाता है. बट जब तक आप डिटेल में क्यूबीडी का डाटा नहीं देते, योर कंपनी विल नॉट गेट परमिशन because QBD involves thorough understanding of process through science and risk-based approach. So this is the conclusion. So these are the some of the references which I use. And if you want, you can go to the USFDA website. You can go for the ANDA filing QBD case study for ANDA filing of immediate release product and modified release product. So here I give an example QBD for IR generic and modified. ये क्या रहता है, USFDA क्या करती है, ये पब्लिश करती हो. And because this is the concept new जब था, when for the first time they introduced in 2005, तो कंपनीज का बहुत रेजिस्टेंस था कि नो वन वाज अवेयर. तो इसका क्या करते हैं, वो वर्कशॉप लेते, वर्कशॉप लेके उनको इनवाइट करते कंपनी रिप्रेजेंटेटिव्स को, उनको बताते. सो इट इज नॉट पॉसिबल फॉर यूएसएफडीए टू गो इन वेरियस प्लेसेस इन द वर्ल्ड. तो वो क्या करते कि जनरली ऑनलाइन कंडक्ट करते एंड दे यूज्ड टू पब्लिश दिस गाइडलाइंस. सो नाउ दैट इज then as I said that ICH harmonized tripartite guidelines for products pharmaceutical product development. तो ये मैं पीजी और पीएचडी के बच्चों को बोलना चाहता हूँ, अगर दे वांट टू मेक करियर इन इंडस्ट्री तो गाइडलाइंस को थोरो होना बहुत इंपॉर्टेंट है. बिकॉज मोस्ट ऑफ द टाइम वी यूज्ड टू रिकमेंड कैंडिडेट फॉर इंटरव्यू, और इंटरव्यू में बच्चे फेल हो जाते हैं. देयर एकेडमिक इज वेरी गुड, पर इंटरव्यू में जो एक्सपेक्टेड था रहता है, उनको कुछ ऐसा पूछा तो इमीडिएटली उनका कंफ्यूजन हो जाता है. सो फॉर दैट यू प्लीज, जैसा आपको टाइम मिलता है, रेगुलेटरी क्या गाइडलाइंस रहती है, ये अलग अलग आईसीएच गाइडलाइंस क्या रहती है, सो यू शुड गो थ्रू दैट. सो दिस इज माय email id, mobile, and if suppose Dr. Sarfaraj, if suppose I'm having time, can I show for 10 minutes one case study?
Yes,
If
sir.
time
Sure,
per
sir. Yes, sir. Yes.
time permits, I can show you.
Yes, sir. Continue, sir. Please. Sir, is there any difficulty?
No, no, just it is processing.
Okay, sir. Okay.
Just I will check. If it can
Okay.
start, then only will. So how the this is the case study my 2011 student. So is it visible, sir?
Yes, sir. It is visible.
Okay. Just I will show directly. I will go to the case study part. Yes. Now you can see this is the desired quality target product profile. So white round tablet, what I was expecting, strength 20 milligram, that which I cannot change. The weight of the tablet 200 to 210 milligram, coating uncoated diameter. So slight way, sometime change in the diameter may be possible, thickness change may be possible, hardness. So variability I have given 7 to 8.5, disintegration time 2 to 4 minutes, assay value, and dissolution in 30 minutes, not less than. So these are the QTPP parameter for me. This is the critical quality attribute parameter. So out of that I selected. So I said that, see that, is this CQA parameter? No. So quality target attributes, but here one more is missing. That is the justification is required. So justification is this CQA. Why you are saying it is no? Someone may ask you. You are saying appearance order is not important. Why you are saying it is no? Content uniformity. Why you are saying it is yes? Dissolution, I said that not less than 80% in 30 minutes in 900 ml of 0.1 normal HCl with 1% SLS using USP apparatus 275 RPM. That was the limit. So why you are saying it is a CQA parameter? Composition क्या रखने वाले हो आप? Lactose, microcrystalline cellulose, cross chromal sodium, magnesium stearate. This is the case study of 2011. So number of case studies are there. This is for simplification because other case studies will take time. Overview of relative risk ranking system, low, medium, high. As I said that, so high risk is unacceptable. Further investigation is needed to reduce the risk. It is very, very important. Example, the same thing what we discussed, solid state form, particle size distribution, solubility, flow properties. So now that is, this chart is important. In as per ICH-Q9 guidelines, solid state form, particle size distribution, solubility, flow properties. Then this is the formulation variables, particle size distribution, lactose, MCC level, or you can write it MCC divided by lactose level, cross chromal sodium. This processing steps, milling, mixing, lubrication, compression. This is my experimental results as per design of experiment, 3 raised to 2 full factorial design, that is the 9 trials which we have planned. And here you can see I have taken the lactose in the range of 60, 70, 80. -1 +1, sorry, -1 0 +1. Here I have taken 4 8 12 factor. So total weight may have adjusted to 100 or 200 somewhere, but cross chromolus sodium, that is the 4 milligram weight, this is -1 0 +1 value, and the response in seconds I have given for the disintegration time. Here you can say that when I'm at the high level of the lactose, 80 milligram and 12 milligram of the CCS, the DT is 45 second only. And if you see, it is at a low level of cross carbamylose and low level of lactose, the DT is more. The same thing you can find with the 100% dissolution in less than 30 minutes, and here it is just 70 minutes. So these values, when you do the study, you find. Now the plot, this is known as the 2F1 plot. So I've gone for the concentration. What is the effect of lactose and cross coromolus sodium on the dt, and this is the time in seconds. So you'll find that that is here from 60 to 80 and 4 to 12 I have given. Now here you can find when I use the cross coromolus sodium in 4 milligram, the disintegration time required is more. But when I gone for the concentration of superdisintegrity, it is more. So the dt, corresponding dt, if you see, it is near about 250. Now individual I can check what happens, and a mesh like structure which you are finding. So this is the interaction. Interaction of what? The lactose. The value of the lactose 70 milligram. What can happen if suppose this is 70 value and 8 value? So this is the mid value where the disintegration time can come. So this is just for understanding how this lactose and cross coromolus sodium affect. So this is the equation what I was talking. So you can see that dt, the plus value here, this is the coefficient, it is the fixed value. A is a lactose and B is cross coromolus. Here you'll find that minus sign is given. So minus sign, what it signifies? So minus sign signifies if you go on increasing the concentration of lactose or cross coromolose, it decreases the time for the disintegration time. It decreases the time of disintegration time. That is the negative effect. This is for dissolution in 30 minutes. The same thing you will find that 80 and 12 and from 60 to 110. You can rotate this. अगर आप जाते हो डिजाइन एक्सपर्ट में 14, तो ये ग्राफ आप कैसे भी उसको प्लेस कर सकते हो. नाउ मैंने कैसे ग्राफ एडजस्ट किया यहाँ पे 4 to 12 and 60 to 80 and here 60 to 110, and because of that when the highest concentration of cross coromolose plus lactose is there, so the dissolution time, so that is the dissolution value is approximately 100. Dissolution value is approximately 100. And अगर यहाँ पे देखते हो आप, तो से फॉर एग्जांपल 4 मिलीग्राम मैंने डाला है उसका, सो डिसोल्यूशन इसका है वैल्यू 80 मिलीग्राम यहाँ पे और यहाँ पे 4 मिलीग्राम, ऐसा पकड़ के चलना है इसको. So the values it comes near to 90. यहाँ पे अगर आप आते हो, so now this values, so at this end 4 milligram of cross coromolose and 60 milligram of lactose in one tablet, so dissolution is 70%. इसको रीड कैसे करना है मैं आपको बता रहा हूँ कि हाउ टू रीड. बच्चों को समझ में नहीं आता मोस्ट ऑफ द टाइम, ये प्लॉट तो दिख रहा है बट इसका रीड कैसे करना है उसको. इसमें मैंने इक्वेशन दिया है देखो कि ये प्लस प्लस प्लस, तो ये जो ए और बी है ये प्लस वैल्यू, इसका मतलब मैं अगर लैक्टोज का कॉन्सेंट्रेशन बढ़ाता हूँ, क्रॉस coromolase को बढ़ाता हूँ, तो इट विल increases the dissolution percentage drug release or dissolution of this. So now that is from the DOE study, it was found that the lactose in concentration of 70 to 80 milligram and cross coromolase sodium in the concentration of 8 to 12 milligram showed DT in less than 4 minutes and more than 80% dissolution in 30 minutes. Now this is my रीजन रेंज. तो मैं कितने कॉन्सेंट्रेशन में, मुझे मेरा टारगेट क्या था कि dt शुड बी लेस देन 4 मिनट्स, डिसोल्यूशन मोर देन 85% और 80% इन 30 मिनट्स. तो इसके लिए मैं काम किया, मैंने ये स्टडी में अगर हमने काम किया जो अभी मैं बताया आपको, सो आई फाउंड दैट in between the range of 70 to 80 milligram of lactose and 8 to 12 milligram of cross coromolase sodium can give me the desired response. Now this is the design space data. So you just please go through. So now that is, this is again for another, this is for my one of research work related to the solid lipid nanoparticle. So I given this data for solid lipid nanoparticles. देखिए सॉफ्टवेयर तो पूरा का पूरा दे देता है बट इंटरप्रेट तो आपको ही करना है और उसके लिए उसको सीखना है. बट सॉफ्टवेयर इस इतना अच्छा है वो सॉफ्टवेयर कि वहां पे क्वेरीज डालते हो तो वो खुद बताता है आपको कि क्या है उसका और उसको इंटरप्रेट कैसा करना है. इसमें मैंने दे दिया था इंटरप्रेट एफिशिएंसी एंड पार्टिकल साइज, इसका टाइम ऑफ स्टरिंग एंड स्टरिंग रेट, ये दो वेरिएबल्स थे फॉर मेकिंग द सॉलिड लिपिड नैनो पार्टिकल्स. सो कंट्रोल स्ट्रेटेजी के बारे में बात कर रहा था. सो कंट्रोल स्ट्रेटेजी में ये देखिए, फॉर इनिशियल मुझे समझ में आ गया कि मेरी रिस्क है तो रिस्क क्या है कि सॉलिड स्टेट फॉर्म डिसोल्यूशन लो, डिसइंटीग्रेशन टाइम आई वांट टू कीप इट एट लो लेवल, तो हाउ कैन आई कीप, जस्टिफिकेशन दे दीजिए, अमॉर्फस फॉर्म was used as it is more soluble than crystalline form. So amorphous form of the drug has to be preferred. Decrease in the particle size. So particle size कम रहेगी तो उसका डिसोल्यूशन ज्यादा रहेगा. तो when I will work, so I will use amorphous form and the less particle size. Particle size distribution contain uniform. तो क्या करना है, improve the flow property of drug which can adjust contain uniformity, dissolution and disintegration. तो उसके लिए उसको उसमें ग्लाइडेंट ऐड करना है आपको. इसके बाद ये जो दिया है सोल्युबिलिटी, अगेन रिडक्शन इन पार्टिकल साइज, इंक्रीज सरफेस एरिया, इंप्रूव सोल्युबिलिटी. तो इसका जस्टिफिकेशन मांगते हो, आपने क्या किया है. फ्लो प्रॉपर्टीज के बारे में मैंने दिया है, ड्रग सब्सटेंस पार्टिकल साइज डिस्ट्रीब्यूशन, माइक्रोक्रिस्टलाइन सेल्यूलोज, लैक्टोज, सो इंप्रूव द फ्लो प्रॉपर्टी. इसका रेशियो जो है एमसीसी टू लैक्टोज रेशियो इज इंपॉर्टेंट टू हैव द कंटेंट यूनिफॉर्मिटी. So improve the flow property of lactose and MCC to improve the content uniformity. Again, the glidant use. Appropriate quantity of CCS can adjust the DT and dissolution. Reduce the level of magnesium state to adjust the dissolution and DT. ये ज्यादा हो जाता है तो हाइड्रोफोबिक हो जाता है, डिसोल्यूशन कम हो जाता है. मिलिंग प्रोसेस, मिक्सिंग प्रोसेस, तो ये जो है वो कंट्रोल स्ट्रेटेजी का पार्ट आ जाता है. एंड बेस्ड ऑन दैट स्टडी फाइनल कंपोजिशन मैंने जो बताया, ड्रग का डोज तो मैं चेंज नहीं कर सकता, बट लैक्टोज प्रोवाइडेड 70 to 80. Microcrystalline cellulose is not significantly affecting to my study. I kept it in 30 to 40. Cross cover 8 to 12. Magnesium 4 to 8. Talc इतना. Total weight of the tablet is adjusted to 180. तो इसमें अगर मुझे 75 मिलीग्राम यूज करना है लैक्टोज को, और मुझे माइक्रोक्रिस्टलाइन सेल्यूलोज 35 यूज करना है 30 की जगह पे, तो मैं कर सकता हूँ, मुझे परमिशन की जरूरत नहीं है, बिकॉज क्यूबीडी का डाटा ऑलरेडी मैंने सबमिट कर चुका हूँ. एंड बेस्ड ऑन दैट क्यूबीडी डाटा नाउ रेगुलेटरी एजेंसी इज कॉन्फिडेंट कि आपने ये पहले स्टडी कर चुके हैं और उसका डाटा ही ऐसा ऐसा है. सो वॉट एवर चेंजेस जस्ट यू ड्रॉप वन मेल, प्रोडक्ट का कोड दे दीजिए, उसके लॉगिन में जाइए और उसमें बता दीजिए कि ये हम ये बैच से हम इतना चेंज कर रहे हैं. दे विल ओपन योर फाइल, विल चेक यस इट इज विद इन द regulatory flexibility range, so they will not ask additional data to you. So this is the post approval changes what we are saying. इक्विपमेंट एंड प्रोसेस पैरामीटर्स में अगर बात करते हो तो बॉल मिल, ये भी हमने काम किया था. तो मिल का स्पीड कितना होना चाहिए, कितने टाइम के लिए, मिक्सिंग क्या है, रैपिड मिक्सर ग्रैन्युलेटर कितना टाइम के लिए, वी शेप ब्लेंडर उसका स्पीड कितना होना चाहिए, कॉम्प्रेशन 12 स्टेशन रोटरी प्रेस 8 mm पंच प्रेस स्पीड. तो ये अगर ये थोड़ा सा हमने इनिशियली ट्राई किया था, अभी उसमें बहुत एडवांसमेंट किया कि स्टडी में. तो यू योरसेल्फ कैन डू दिस स्टडी. सो जो रिसर्चर्स है वो कर सकते हैं, फैकल्टीज भी इसके रिलेटेड काम दे सकते हैं. एवरी टाइम इट इज नॉट पॉसिबल फॉर अस टू गो फॉर द प्री क्लिनिकल स्टडी, सेल लाइन स्टडी, बट इफ यू कैन सपोर्ट योर डाटा विद द क्यूबीडी दैट इज आल्सो एक्सेप्टेबल. So this is from my side.
Thank you, sir, for such an informative session and sharing your expertise with us. Thank you, sir.
Yes.
So
thank you, sir.
So due to some technical issues, today, the coordinator of our program, Dr. Amir Kaji sir, was unable to join, but now he is with us. So I would like to request Dr. Amir Kaji sir for sharing some say a few words regarding the session. So good afternoon everyone. Due to some technical issue, I was unable to join the session. So myself, Dr. Amir Khazi, takes an opportunity to welcome you all to one-day national level seminar on application of QBD in preformulation study. Dear participant, QBD is one of the challenging concepts nowadays, which we have already learned from Dr. Mahaparle sir thoroughly, and I hope this will be definitely useful for all of pharma buddies. So we are very fortunate that we have got such an wonderful and eminent personalities for our seminar, Dr. Mahaparle sir, and Dr. Degan sir in the afternoon session for their valuable time and knowledge for all of us. I am also very thankful to our management, our principal, for giving me opportunity to organize the seminar. Thank you one and all. Thank you.
Thank you, sir. Sir, how many participants are there? Kaji sir? सरफराज सर पार्टिसिपेंट्स किती आहेत म्हटलं आता?
Sir, we have more than 50 participants right now.
Okay, sir. Okay.
Yes, yes.
So thank you
with
very much,
your
sir.
permission, can we start the question and answer session, sir?
Yes, sir. Please.
Okay, sir. So we have one participant, Yogesh Patel. So he is asking that how quality is affected by its design, as design is basically for a medication, attractiveness and increasing its efficacy.
No, design is not in that sense. We are using the word design. Design means what? So product का क्वालिटी इम्प्रूव करने के लिए आप जो उसमें एक्सिपिएंट डाल रहे हो, जैसे मैंने बताया कि व्हाट इज द क्रिटिकल क्वालिटी एट्रीब्यूट पैरामीटर फॉर योर प्रोडक्ट, उसको पहले आप आइडेंटिफाई कीजिए. दिस इज वन पार्ट ऑफ द डिजाइन. अनदर थिंग इज दैट व्हाट आर द excipients, what process is going to affect the quality of your product, or the second step of the design. And then third thing is that आप उसकी रेंज, पहले तो आपको क्यूटीपीपी आइडेंटिफाई करना है, उसके बाद सीक्यूए आइडेंटिफाई करना है, रेंज को आइडेंटिफाई करना है, और उसके हिसाब से कौन से एक्सिपिएंट को आपको लेना है. सो दिस इज वन डिजाइन कि बाबा हम ये एक्सिपिएंट यूज करेंगे, ये प्रोसेस यूज करेंगे. उसमें एक्सिपिएंट की क्वांटिटी क्या होनी चाहिए, what should be the quantity of the excipient, the range of the excipient, that is also one part of the design. तो डिजाइन क्या है कि क्वालिटी नॉट कम्स बाय ड्रीम, क्वालिटी नॉट कम्स बाय ड्रीम. यू हैव टू गिव इनपुट. अगर आप इनपुट देते हैं इन प्रॉपर वे, इफ यू आर गिविंग द इनपुट, देन ओनली यू कैन गेट द डिजाइन. अगर मुझे कुछ एक स्टैचू बनाना है, कुछ करना है, तो उसको मुझे कुछ ना कुछ तो एक डिजाइन चाहिए, एंड समथिंग शुड बी इन माय माइंड कि मैं क्या बनाना चाहता हूँ. ऐसा नहीं रहता कि यहाँ ठोक ठाक के उसको बनाया जाएगा. सो इट शुड बी इन माय माइंड कि आई वांट टू मेक दिस, so I want to make this product which is having this quality, and how can I get this quality? For that, the designing is important. I think it is clear. I hope.
Yes, sir. Thank you, sir. So there is one more question from our participant. What is the role of QBD in clinical trials?
This QBD can be used in a clinical trials. As I said that not only it is restricted to this. So clinical trials में आप अगर जो volunteer selection करते हो आप, तो उसके volunteers का जो रहता है, उनका मेल्स है, फीमेल्स है, उनका एज ग्रुप क्या है, राइट? तो किसके लिए कर रहे हैं आप स्टडी, फॉर व्हाट पर्पस यू आर गोइंग फॉर क्लिनिकल ट्रायल्स. सो ये ड्रग का इफेक्ट चेक करना है आपको. तो उसके लिए वॉलंटियर जो रहेंगे आपके, तो वॉलंटियर्स का उसका विच वॉलंटियर्स वी कैन सिलेक्ट, उनका एज ग्रुप क्या रहेगा उसका. सो नाउ दैट इज वेदर दीज आर द हेल्दी वॉलंटियर्स और यू आर गोइंग फॉर द पेशेंट. सो ये आपको डिसाइड करना है कि बाबा ये चीज हम फिक्स करेंगे, और उसके बाद व्हाट यू आर एक्सपेक्टिंग, कि रिजल्ट क्या एक्सपेक्ट करते हो उसको, व्हाट आर द रिस्क एसोसिएटेड. अगर ये मेरा डोज कम ज्यादा हो जाता है तो व्हाट कैन हैपन टू द पर्सन, so that risk is associated. So there we need to focus more, and there we need to keep the control. उसमें कंट्रोल अगर हम करते हो तो that is also one part of the QBD, quality by design. तो मैंने जो बताया है कि इट इज नॉट ओनली रिस्ट्रिक्टेड, फार्माकोलॉजी में जाओगे वो हो जाएगा, कोग्नोसी में हो जाएगा, क्लिनिकल ट्रायल्स में जो हो जाओगे, so there also we required the quality by design.
Okay, sir. Thank you. We have one more question, sir. What are the applications of QBD in current drug development?
So drug development, if suppose in drug discovery from medicinal point of view, medicinal chemistry point of view, so you will see there also it is having the application. And in drug development is comes under the part of drug discovery, and product development comes under the formulation and development. So at both the places, so quality by design is having the importance. अगर आप प्लानिंग कर रहे हो कि मैं डॉकिंग स्टडी करता हूँ मोलिकुलर डॉकिंग करता हूँ, फिर उसको स्क्रीनिंग करता हूँ, देन आई यूज टू गेट दिस स्ट्रक्चर आई वांट टू मेक एज अ एंटी हाइपरटेंसिव मोलिक्यूल, तो उसको स्क्रीनिंग किया ना, आपने स्क्रीनिंग किया, इसका मतलब आपने उसको डिजाइनिंग करना चालू कर दिया। सो क्वांटिटी जो क्यूएसआर डिजाइन ही बोला जाता है उसको, सो ड्रग डिस्कवरी में आ जाता है। अभी वो मोलिक्यूल आपके हाथ में आ गया फॉर्मुलेशन डेवलपमेंट के पर्सन के, just wait about 2 minutes. When you will work, आपके हाथ में मोलिक्यूल आने के बाद आपने क्या किया, उस पर काम करना चालू कर दिया। सो उसका डेवलपमेंट करना चालू किया, प्रोडक्ट डेवलपमेंट। सो देयर आल्सो वी रिक्वायर्ड टू हैव द क्यूबीडी। सो एट बोथ द प्लेसेस क्यूबीडी इज हैविंग द एप्लीकेशन।
Okay, sir. Thank you. Now, I would like to request Mrs. Rajmane Madam to give vote of thanks.
Am I audible, sir?
Yes madam, you are audible.
Thank you, sir. A very good afternoon to all of you. On behalf of Adarsh Shikshan Prasad Medical College of Pharmacy and on behalf of all participants, I am Ms. Ragini Rajmane, glad to propose a vote of thanks for this first session of one-day seminar. My heart fills with lots of gratitude and respect for our today's knowledgeable and eminent personality and the speaker, Dr. P.R. Mahaparal sir, not only for sparing their valuable time and knowledge with us, but also for enlightening us with their admirable talk on this important topic, and also for clearing our concepts and enhancing our understanding pertaining to the topic applications of QBD in pre-formulation study. Thank you, sir, for such a nice and in-depth session. Lastly, I must say the session was very informative and helpful for upgrading our knowledge related to the said subject. We will be always grateful for your kind cooperation for success of this seminar, sir. We are very fortunate to have you as a research person. Thank you so much, sir.
Thank you very much madam, thanks.
Thank you. I would like to present certificate of appreciation to respected Dr. Mahaparai, sir. Please accept, sir.
Yes sir, thank you very much madam, thanks.
Thank you. Thank you, sir. Now, there are some instructions for participants. There is a end of this first session. There will be break for 1 hour, and then second session will be start sharp at 2:30. So have a lunch and join again at 2:30. Secondly, for issue of e-certificate, it is compulsory to attend second session and it is compulsory to fill the feedback form. The feedback form link will be shared in the afternoon session. So please do attend the second session. So now, with the due respect, we would like to conclude the first session. Thank you, sir. Thank you, all the participants.
Thank you madam, thanks to Principal Joshi sir, thanks to our Kaji sir, Dr. Sarfaraj, and all your team, faculty members and management for inviting me, for giving an opportunity to deliver my talk. And thanks to all participants patient listening.
Thank you, sir.
Thank you. Can I
Thank you,
quit,
sir.
madam?
Yes, sir. Yes.
Okay. Thank you, madam. Thank you.
Thank you.